At Precision, we have a proprietary gene editing platform that we call ARCUS. It's distinct from CRISPR in a number of different ways, although many people have heard of CRISPR. So it's a good place to kind of start the conversation. ARCUS is unique in really three fundamental ways that we think are important when you think about developing therapeutic gene editing programs. One is the way in which ARCUS actually cuts the DNA. It cuts it with a unique type of cut that in this particular program allows for perfect religation of two ARCUS cuts. So we use two nucleases to cut out a region of the dystrophin gene, and because of those overhangs, it actually comes back together perfectly the vast majority of the time, which is really, we see a good safety feature of the approach. The other way that ARCUS is really different is it's very small. So it's an incredibly small editor, the smallest that you've probably seen. We can fit two ARCUS nucleases in a single AAV, and so our approach for DMD is to actually use two ARCUS nucleases packaged in a single AAV to excise that region of the dystrophin gene. So size is really a key advantage when thinking about applying gene editing technologies therapeutically because it has really massive delivery implications. And then finally, ARCUS is unique in that it's one protein that can bind the DNA and cut the DNA. And so you don't have a guide RNA. It's not multiple components needing to get to the same place at the same time. It's one protein that can bind DNA and cut DNA. So again, simplifies delivery, and we think using our protein engineering skills, we've allowed those two nucleases to work together well, coordinated cuts to allow for efficient excision of that region in the dystrophin gene. So really very different in a number of important ways from CRISPR technologies, but that's what we do at Precision. We develop ARCUS nucleases for therapeutic gene editing.
Have you guys had any conversations on what a first in-human study would look like with this sort of gene editing platform, or what that would entail possibly?
Yeah. I want to comment first on the durability question because it's something that we really kept in mind when we designed this approach. And it's something that I think I want to be sure to sort of emphasize with using an AAV vector. And so we are using an AAV vector, but the payload is different than a microdystrophin approach. So in our AAV, it's encoding ARCUS nucleases that go in and correct the gene in the DNA, in the cell, in the patient's own body. That means that we're not dependent on persistence of the AAV long-term for continued effect. The edit happens quite fast, and then if the AAV were to be silenced or diluted out over time, the edit persists. It's a permanent edit at the DNA level. And I think contrast that with a microdystrophin approach where the therapeutic effect is being delivered in the context of an AAV vector. And so if that vector is silenced or lost over time, the durability of the effect may be limited. And so we really designed this approach with durability in mind, with permanence, long-term efficacy in mind. In terms of what that first in-human trial could look like, yeah, that's a major goal of ours here at the MDA conference is working with clinicians in the space who have a lot of experience treating DMD patients to understand what a successful trial could look like. I think we are trying to learn from those who've gone before us in terms of what are the right endpoints, what are the right inclusion criteria. It's too early, I think, to really say exactly what our trial will look like, but we are actively working hand in hand with the physicians in the space to really smartly design this trial.